Hello, good morning. We're gonna get started. We will be doing the test one review today. I know you guys have a lot of evaluations this week in addition to the biology one. So I will try to keep it brief. You already have the review notes. Sorry, the review package. So, hopefully you've printed that out. And we are basically just going to go through the question. I'm going to be using a lot of space here, so I will not turn my camera on today. You don't need to see me. We'll just try to, utilize as much of the screen as possible for the review. Okay, so first question here. What is the control of the experiment? Talking about the beetroot question, beetroot lab. There will be a few beetroot questions on the test, so make sure you familiarize yourself with these ones. So the control, right, is the room temperature, right? Room temperature test tube, right? Many of you got this, but some of you set the 0-degree temperature, or they say some other ones is not. Like the control is the one that you do not change, right? You do not manipulate. There's no treatment to it. There's no modification done to it. So the room temperature test tube is the control. And the purpose of the control is to provide a reference to compare with. Okay? Without the control, how do you know what normal looks like? You don't. That's why you need to have a control. You need to have a reference to compare all the other test tubes to. Okay. Next one. What is the dependent and independent variable? Dependent variable is going to be the color intensity. Okay. Or you can say absorbance. Right. And then we will have the independent variable. Independent variable is going to be the basically temperature, right? Temperature. Okay. What variables are standardized? So these are things that are kept constant, right? So for example, we have size of the beetroot, discs, right? Like when she cuts it up into little pieces, you want to make sure they're the same size. Like otherwise, bigger ones is going to give off more color, right? So we want to standardize that. The time spent in water bath, you want all of them to be 20 minutes long. If it's too long, then all of them might reach the same color. If it's too short, they might not reach the temperature that is required, and the color will be underdeveloped. Okay. Anything else? Standardized variable? Does anybody have any additional ones? Okay. Volume of water in each test tube. If you add too much water for some of them, you might dilute the color. If you add too little, then you might, concentrate too much. Okay. Next, why does the frozen food has similar color intensity as the 80 when placed in water at room temperature? This one I talked about to you in class, right? the frozen beetroot, basically the ice crystal damage the cell membrane. So when you thaw it at room temperature, everything just leaks out because the membrane is already damaged. Okay, so that's why like when all the color leaks out, it's as if the membrane is very permeable. What do you expect the absorbance to be if we include temperatures higher than 80 degrees? So if you look at your graph, and a lot of you did the right graph, it should be a line graph, right? There's a rise here, and then it goes like that, right? So the peak occurs at around 70 degrees. Afterwards, it's just kind of flat, right? Like there's no, it's not rising anymore, right? That's why this is the peak, and this is temperature. Sorry, this is absorbance, and this is temperature. And so if we heat it up any further, absorbance should remain unchanged, right? Because at 80 degree, the absorbance is already peaked out. The cell membrane at that point is completely broken down already. Right? The membrane protein has denatured, and you basically unravel the entire membrane. All the pigment has leaked out. Right? So even if you heat it up higher, you're not going to be able to extract more color from it, right? So it would be no change. No change. Okay, so that's all you have to know for the Beetroot Lab. Any question before I move on to lecture 1? All right. What is a placebo? Give an example of an experiment where a placebo is needed. So a placebo is basically a sham treatment, right? Sham treatment. It is something that has no effect on the dependent variable. Okay. And the purpose of a placebo is to eliminate bias or minimize, we can say, right? Minimize bias. Okay, and you need it when you deal with human subjects. Okay, needed for experiment involving humans. Okay, give an example, another example where placebo is not needed. So like testing with animals, right? In class, we had an example where you want to see if increase protein in diet for cows equals to increase milk production, right? That was a question. And in such case, you don't need to have a placebo. Right? You just give them one group, you give them proteins, high proteins. The other, you give them regular proteins. Cows don't have biases. So that's what a placebo is. Now moving on. What is a valid hypothesis? A hypothesis should be testable and falsifiable. All right. Practice writing a hypothesis. So you did quite a lot of that in the experiment, in your worksheet. Just make sure that your hypothesis is not a question, and make sure that the hypothesis, you don't have to explain things. So for the Beetroot Lab, right, the hypothesis would be increasing temperature will increase membrane permeability. That's it. Okay, you have to be specific, right? Don't just say increased temperature will affect the membrane permeability. How is it going to affect it, right? Is it going to increase it? Is it going to decrease it? That's you have to be specific, right? All right, look at the test results below for testing the efficacy of the sleeping pill, right? To reduce jet lag symptoms. So which one shows that it is effective? So let's take a look here. We have the control group. Both placebo and the drug were able to reduce the jet lag symptoms, right? So in this case, both placebo and the medicine, the drug, reduce jet lag, right? Therefore, does not work, right? Because if the placebo works as good as the actual drug, why do I need the drug? Why don't I just take the placebo, right? This is telling you it's mostly a psychological effect. Next one. Placebo is similar to control, and the medicine is able to reduce, right? So only the drug reduces symptoms, right? Therefore, effective. Okay. Now there are other things you should know for test one. This is not like an exhaustive list of everything you have to know, right? It's just like some review question. So other things you should know would be like the experiments, right? Rady and Pasteur's experiment. Okay, so if you need to refresh your memory, go through the video archives that we have. With the flies, which one is control, which one is independent, so on and so forth, right? Okay. Any question from lecture 1, scientific method? Okay. In terms of difficulty, it's not more difficult than the quiz. It's just like more information, right? The format is very similar to what you have seen so far. Yes, the test is on Thursday. There will be no class on Thursday. You just take the test. Okay, it's gonna be available to you from 6 a.m. to midnight. All right, lecture 2 then. What holds the atoms together in water molecule? That would be a covalent bond, okay, which involves the sharing of electron. How would you describe a water molecule? Well, overall it is neutral, okay, but it's polar. Polar means part of, right, the oxygen is slightly negative, right? Whereas the hydrogens are slightly positive. And that is as a result of uneven sharing of electron, right? We talked about this concept of electronegativity last time when we described the ETC. So oxygen is more electronegative, right? That's why it's pulling the electron towards itself more. Yes, the notes will be posted afterwards, as usual. Okay. So what interactions exist between two adjacent water molecules? Well, if I put an H2O next to another H2O, since this is slightly positive and this is slightly negative, they will be attracted to each other, right? And this attraction is called a hydrogen bond. Hydrogen bond. Okay? So we've seen hydrogen bond at various places. Right? It's not just something that exists between two water molecules. Can you guys give me an example of another molecule where we discussed hydrogen bonds? That's okay. For example,
NaCl dissolved in water.
oh, okay, I see what you mean. That's talking about hydrophilic compound being able to dissolve in water. Not quite hydrogen bond though, but thank you for trying. I was thinking about DNA. The two strands of DNA are held together by hydrogen bonds, right? The A's and the T's, they have two hydrogen bonds, and then the G's and the C's, they have three hydrogen bonds, right? We also talk about hydrogen bonds holding together secondary structures in a protein. Okay, so maybe I'll write down for you here just in case you need it. So hydrogen bond, not related to water, but hydrogen bonds exist in proteins and DNA, right? So it holds together the two strands of DNA and it stabilizes secondary and tertiary structure. Hey, is your math midterm today or on Thursday as well? Just curious.
Thursday.
Thursday? Okay, thank you. Moving on. If you supply radioactive oh, some of you have it today. Interesting. If you supply radioactive nitrogen atoms, something that can be traced and detected to a cell and let it grow, what molecules would you expect to find radioactive labels in? Okay, so this is basically a question that asks you, where do you find nitrogen atoms in a cell? Okay, so nitrogen atoms, they are found in lipids and proteins, right? Sorry, not lipids. Proteins and nucleic acid. Proteins and nucleic acid. Okay, remember proteins is made up of CH and O and then trace of sulfur. Nucleic acid is CHNOP. Right, so both of them has nitrogen. You would expect these molecules to pick up the nitrogen atom, the radioactive nitrogen, and you'll be able to identify them. What is the monomer of DNA? Monomer of DNA is a nucleotide. Nucleotide has three parts. There is a phosphate. There is a deoxyribose in this case, which is a sugar, and then plus a nitrogen base. Right? Because we're talking about DNA, the four nitrogen base that are possible would be A, C, T, and G. How are nitrogenous base classified? So we have the purines, which are two rings, and they include A and G. And there are the pyrimidines, which are single ring, and that includes the T and the C. Now, hopefully these are not unfamiliar to you, right? We've learned these before. This is just a review sheet. What is the base pair rule for DNA? Well, purine pairs with pyrimidine. Okay. Specifically, A pairs with T and C pairs with G. All right. What force holds the nitrogen bases together along the length of the DNA? Well, I told you earlier, right? It is the hydrogen bond. Hydrogen bonds, right? 3 bonds, 3 hydrogen bonds between C and G, and 2 bonds between A and T. Right, so it's much harder to pull apart a strand that has a lot of C and Gs, and then easier to pull apart strands that are only A's and T's. What is the significance of base pairing rule? Well, base pairing rule means the C and G and A and T, right? Because we're always pairing a purine with a pyrimidine, so the width is always going to be 3 rings wide. Okay, so this ensures consistent width across the length of the DNA. Okay? It's always the same width, which increases stability, right? Okay, so I'm just gonna pause for a second and let you catch up. Next, what is dehydration synthesis? Now you should know that by now, right? Dehydration synthesis is when you basically take 2 molecules, okay, 2 molecules, and each of them has an OH. Right? And then what you can do is you can remove an OH from one and remove an H from the other. So in the end, you take out a water, but more importantly, you're able to create a link, right, that connects them together. Okay, so dehydration synthesis basically connects monomer together to form polymers. Right? Regarding enzyme, what is an enzyme? Enzymes are proteins, right? Proteins that catalyze chemical reaction. Okay. They speed up reactions in the body, essentially. And how does it work? Enzyme works by lowering activation energy, right? So there's the graph that I keep on showing you, right? Okay, this is energy, this is time, this is the original activation energy. Right? It's very high, but if you have an enzyme, you will have the same reaction, but this time you have a lower activation energy. Okay. Now, we also describe how the enzyme has these pockets, right? So these are called active sites. And then you would fit your substrate in, right? Substrate. And then the enzyme will essentially carry out the reaction. It could be different kind of reactions. You could be breaking the bond, right? Breaking the bond in the substrate. That's hydrolysis. It could be like what we learned in lecture 5, the substrate level phosphorylation. Maybe it's transferring a phosphate from one thing to another, right? So the enzyme has an active site that allows the substrate to fit in. And different substrates have different shape. So they would only be able to fit into a particular enzyme. Next, what factors affect the functionality of an enzyme? Well, since an enzyme is made of proteins, so anything that alters the shape of the protein is going to affect its function. So example, like temperature, right? pH, ionic concentration, right? All these things are going to affect the enzyme, right? By altering the shape of the enzyme, basically, right? So anything that affects the shape, right? Shape is also called conformation, right? Of a protein. What does the enzyme lactase do? Right? Anytime we have something that ends with ASE, it's likely to be an enzyme. And lactase basically breaks down lactose. Right? So we have galactose plus glucose, which makes lactose. Now, if you want to break down the lactose, let's say you ingested some lactose, some dairy product, lactose, if you want to break it back down into galactose and glucose, you will need to have an enzyme to do that. And the enzyme is lactase. Right? I told you about people who are lactose intolerant. They could buy lactase at pharmacy and take the pills before they eat their ice cream. Artificial lactase. Okay. Hope that makes sense. Next, we have a chart for each of the macromolecules that we've learned. Let's go through it together. starting with carbohydrates. The monomers of carbohydrates, well, we have a bunch, right? There's glucose. There is galactose. And there's also fructose. Right? So these together, all of them collectively are known as monosaccharides, right? Monosaccharide, single sugar. Now you can, of course, put these together. I'm not going to write them out again, but then glucose plus glucose gives you maltose, right? Glucose plus fructose gives you sucrose, and glucose plus galactose gives you lactose, right? And still, you can make them even longer. And when you do, right, you get the polysaccharides, right? So biological functions, right? Main source of energy. We spent a whole lecture talking about how our body metabolizes glucose to produce ATP, right? So carbohydrates is made up of glucose. That's the main source of energy. Storage form of energy. And also, there is structural support, right? Let me see if I can make this chart table. Oh, there we go. Structural support. Okay. So examples. For storage, we have starch in plants, in the roots and the stems. And humans, we can also store energy in the form of glycogen, right? In the muscle and liver. Okay? Starch in stems, stem and root, glycogen in muscle and liver. Okay. Structural support. Anybody remember examples of polysaccharides that provide structural support in plants and some animals?
Cell wall or membranes.
That's right, the cell wall, right? And what is the cell wall made up of?
Cell membranes.
No, the cell wall is different from the cell membrane, right? Cell wall is
Oh
outside of
yeah,
the cell membrane, yeah?
yeah, yeah.
Yeah, but what is it made up of? Anybody remember?
Fiber.
Fibers, okay, yeah. And the fiber is made up of? We're getting there, getting closer. That's all right. Cellulose, right? Cellulose. Oh, thank you, Sarah. Cellulose for cell wall. And we also have chitin, right? Chitin for like shells, right? In crabs, lobster, clams, bugs. Okay? So these are polysaccharides. Okay, polysaccharides. Okay. So that's all the carbohydrates. All right. I know it's a lot to remember, but you still have time. Next, we have proteins. Proteins. The monomer for protein is amino acid. Amino acid. Okay. So amino acid looks like this, right? You should recognize an amino acid when you see one. you don't have to draw it, but you have to recognize it, right? So there is the carboxyl group. There is the amino group. And then we have a side chain. So all the proteins are made up of 20 amino acids. And all the amino acids share this structure. The only thing that separates one from the other is the kind of side chain that it has. Biological functions, we have lots. For example, they form membrane proteins, which plays a role in structural integrity, transport. Well, we have 6 of them, right? Remember? Cell recognition, forming tight junctions, so on and so forth. We'll review those later. Proteins are also important for structure, like muscle, nails, hairs. All these things are structures, right? And they are made up of proteins. We also have proteins that will form hormones. Okay, some hormones are lipid, some hormones are proteins. Okay. So, of course, there's energy source. Right? We learned last class that the proteins can be broken down into amino acids, and depending on which amino acid it is, you can feed them into different parts of cellular respiration, right? Okay. So let's give some examples. Membrane proteins, right? So, for example, like transport, right? So we learned 2 types of transport proteins in the cell membrane. Does anybody remember what they are?
FADH, NADH, FADH, NADH.
Oh, those are not proteins, though. Those are electron carriers. But again, thank you. Thank you for trying. Anyone else?
Enzyme?
Yeah, okay. Enzyme, ribosomes, okay. No, not quite. Anyone else? Oh, you guys are worrying me. The test is in 2 days. Okay, let me rephrase the question. We talked about membrane. We talk about transport, right? That's moving things across the cell membrane, right? So when things are too big or when things are charged and they need to get across the cell membrane, they need the help of transport protein. And there are 2 types that we talked about. What are they?
Integral canal.
Yeah, we're getting closer. Yeah, so they are integral proteins that plays a role in passive transport, perhaps. No, should I just tell you? Okay, I'll tell you. Carrier protein, right? Or channel protein, right? Does that ring a bell? We rolled this whole note, right? On I think it was like a Thursday morning, right? With the flow chart. Let me show you to jog your memory. That was lecture 4. Like this whole table, right? Remember? Right, so like channel protein, carrier protein, right? These are membrane proteins that plays a role in transport. Is that okay? Does that ring a bell? Hopefully ring several bells. Structure. I talked about structure already, right? Like muscle, right? Hair, nails. Hormones. like insulin. Okay. And of course, I missed one here. They can also be enzymes, right? And we have the lactase. Okay. All right. So lots of work you guys need to do. For the proteins, of course, there's also the different levels, right? The primary, secondary, which we'll review in a second, I guess. But let's move on to lipids for a second. There are no monomer for lipids. Instead, they have building blocks, right? Okay, they have components. So glycerol, fatty acids, which of course can be saturated or unsaturated. And you can have like phosphate groups, right? Phosphate head, carbon rings, right? And these are the main components that we've learned. In terms of functions, storage of energy, excellent way to store energy. Energy storage, insulation, and protection. I'm sorry, what is it? Did someone say something? I heard no. Okay. Insulation, right? And major component of cell membrane. Okay. Hormones as well. So in terms of like major component of cell membrane, that would be the phospholipid. And also cholesterol, right? Cholesterol helps control the fluidity of the cell membrane. More cholesterol, more fluid. Hormones, anybody? You must remember this, right? Give me example of hormones. Steroids, that's right, steroid hormones. Do we have a specific example of steroid hormones? They are made of cholesterol, but cholesterol is not a hormone itself. That's right, estrogen, right? Testosterone and that kind of stuff, right? That's that. Well, hopefully, this review is helping you, right? Like, I don't know if you guys are just being quiet or like, you're worrying about other subjects. Um, but if you haven't started studying the bio, um, you should probably have this sheet next to you when you do the test, right? Which is fine, right? Like I keep on saying, right? It's okay. That's the advantage of doing this online. Like you can use reference, right? Um, but again, I want to emphasize this is not an exhaustive list of all the things you have to know. This is a good starting point. All right. If you have limited time, then these are the stuff that you should remember. Nucleic acid, the monomer is nucleotide. Okay, and you know, if we're talking about DNA, then it's ACTG. But for RNA, we don't have thymine. Does anybody remember the alphabet in RNA? Instead of thymine, what do we have? Not quite R, but good try. Anyone else? That's right, U. Thank you, Sarah. U for uracil. Okay, and what do we use it for? Well, they are the genetic material, right? They get passed down to the next generation, right? To be passed down to next generation. They have the instruction to make proteins. And we really only have two types, DNA and RNA. Well, you kind of know that there are different kinds of RNA, right? You should know at least there are messenger RNA, which is needed in the process of transcription and translation. And we also have rRNA that makes up the ribosome, right? That's it. How is the shape and structure of a protein stabilized? So there are 4 levels. Let me just briefly summarize it for you. For the primary level, primary is basically the sequence, right, of amino acid. Okay, sequence of amino acid. And what's holding them together are basically covalent bond, right? Covalent bond. And then the secondary structure, they either form these spirals, the helices, or they have these zigzag, right? And so that's like called the beta sheets. Or the alpha helix. The primary structure is connected together through covalent bonds. These are stabilized by hydrogen bonds. Hydrogen bonds hold them together. Right? We already talked about other examples of hydrogen bonds. Hydrogen bond that exists between water molecules, hydrogen bond that holds the ACTGs together in DNA. And then we have the tertiary structure, right? Tertiary structure, which is the overall 3-dimensional shape. Remember the pipe cleaner, right? That little tube that I was trying to show you. Right, so they fold up, 3-dimensional structure, 3D conformation. And this one is held together by many things. So it could be hydrogen bonds again. It could even be ionic bond. Okay, hydrophobic interactions. So essentially, these are all interactions of side chains. Okay. Interactions of side chains. And then you have the quaternary structure. I'm running out of space. I'm not going to write it down. But basically, you have multiple polypeptide coming together, right? Like you might have 2 pink ones plus 2 blue ones, and that forms like a complex. Okay, we call that the quaternary structure. Not every protein has the quaternary structure. Some is fully functional after the 3rd level. Okay, but some do. Some have the fourth level. Now, if you heat it up, if you heat things up, if you change the pH, if you change the temperature, if you change the ionic concentration, this is important to write down. Okay, it will only affect the secondary, tertiary, and quaternary structure. Temperature alone is not going to break your covalent bonds. Okay, pH alone is not going to break the covalent bonds. Okay. Does anyone have any question? Okay. Let's take a look here. Identify the following molecules. This, like I said, you should recognize is amino acid. This one, phosphate. You learn phosphate in chemistry, right? PO4, -3. This is the sugar, the ribose sugar. And then this is the nitrogen base. So tell me, what do we call this whole thing?
Nucleotide.
That's right, that is the nucleotide. Very good. Over here we have a monosaccharide. I mean, you don't really know which one it is. Glucose, fructose, galactose, you don't have to know, but it's a single sugar. And this here, if you see a bunch of C and H together, that is going to be the fatty acid. Okay. Let's take a look at the next one here. List the major components of cell membrane. I mean, we drew a picture of this, right? With the fluid mosaic model. But you know, that's fine. We'll list some key things. Okay, so phospholipid, right? Phospholipids. We have some are saturated, some are unsaturated. Sorry, my writing. Unsaturated. This would make it more solid, and this would make it more liquid. And it's not about, you know, whether one is better than the other, right? It's about the environment that the cell is in. If it's in a hot environment, you want it to be more solid, so it's not going to melt. Right? If it's in a cold environment, you want it to be more liquid, so it's not going to freeze. Okay. Cholesterol, okay, which adjust membrane fluidity. More cholesterol equals more liquid, right? There are proteins. Proteins have various functions. I think you know we might have a question coming up, but in the lecture, do you guys know what I'm talking about? Like, let me see. It was lecture 4, I think. There is this table, right? That talks about all the functions. It's in your PowerPoint as well. So like attachment to cytoskeleton, cell signaling, enzymatic activity, cell junction, right? So on and so forth, right? So these, depending on what kind of proteins you're looking at, they're gonna have various functions, right? Okay, so see lecture 4 slides, PPT, PowerPoint for 6 key functions. Transport, right? Cell recognition, so on and so forth. And you also have something like glycoprotein, sugary proteins, and glycolipids. And these are for cell identity, right? Allows your cell to distinguish between, you know, what belongs to the body and what does not, right? Okay. Central dogma of biology. Central dogma of biology. That is starting at the DNA. You're gonna basically make a copy of the section that you need, and that is called the transcription, right? And then once you have that copy, you're gonna use it to make the protein, and that is called translation. Right? Transcription happens in the nucleus, whereas translation happens in the cytoplasm. Right? So that involves ribosomes coming along, right? One of the functions of liver is to remove toxins from the blood. Based on this, which 2 organelles would you expect to be abundant in liver cells? Well, we have things like smooth ER for detoxification, right? What other organelle is capable of detoxifying? Anybody? And other organelles that can detoxify. Lysosome. Right? So both of these will be abundant, right, to help with the breakdown. All right, why don't we take a 5-minute break and then we'll do more. I'll give you some practice questions afterwards as well. So don't go away, but you know, do take a break. See you soon. You said you would put the poll question. Yeah, I put it on already. Is it not there? Let me see. Let me see if it's there. Just give me a second. So if you look at the announcement, right, it says I also put all the poll questions in the PowerPoint file. Just click on it. I guess the link is really tiny, right? Maybe some of you missed it. If you click on that, there it is. Okay, you can't see the answer here, but after you download it in the PowerPoint, the answer is like at the bottom. Okay. If you still can't find it, just yours doesn't show. What do you mean? Like when you click on it, the file is blank or something? It says file not found. Does anyone else experience the same problem? Same? Okay, let's see. You guys are 201, right? Test review. Oh, it's my problem. I'm sorry. I forgot to make it visible to students. Would you mind trying it again now and let me know if it works? Probably did the same thing for my other class. Let me see. It works now? Okay, yeah. Sorry about that. Thank you. Thank you for letting me know. Sometimes I just, don't pay attention as much as I should. Okay, well, now that we got that sorted out, make sure you have those poll questions with you, right? 'Cause by now you know they're gonna show up. Some of them, anyways. Okay, describe the structure of a mitochondria and state of function of each part. I mean, we did this many times already. And by now, after lecture 5, you should be very familiar with the structure of the mitochondria, right? But for old time's sake, let's go through it one more time. This is the outer membrane. We have the inner membrane. Which forms the cristae. And yes, of course, there's the matrix on the inside. And the intermembrane space. And we know more now than we did when we first learned about this. Now we have the ETC here, right? All these little proteins here. Right? Those are the ETC. Describe the endomembrane system. So, long story short, we basically have the nucleus here. Okay. And I show you this before, that the nucleus has an envelope, right? A nuclear envelope that goes around it. Oh, by the way, like next time, if any of you find something that's not working on eCentennial, like if there's a broken link or something or file missing, just send me an email. I'll fix it right away. Okay, you don't have to wait until the next class. And then this is gonna extend, right? Outside into here. Hi, and then we're gonna have some different organelles here. So basically the endomembrane system involves the rough ER, right? That's the rough ER. It's gonna produce a protein and package it in a vesicle. This goes to the Golgi body, where it will repackage it into a new vesicle, right? It sorts the content, it modifies the content, and then it repackages it. And then this can go various places, right? So I'm gonna say C, lecture 3 notes. I'll verbally go through them with you though. It could leave the cell, right, through exocytosis. We know that by now. It could become part of the cell membrane. We know that there are membrane proteins. It could stay in the cytoplasm until it's needed, or it could stay in the cytoplasm to be used within the cytoplasm. Or it could become a lysosome, right? Lysosome. Okay, so obviously there are a lot of organelles that exist in the cell, and we only cover some of them. So make sure you go through them yourself and you know their functions. Yeah, let's try this question. Oh, why is it not moving? Oh, there it is. Do we need to explain it? But no, there's no word, there's no written response question on this test. Okay, so everything is multiple choice. But I mean, you got to understand this concept, right? In order to answer some of the questions. I can say you're like, where do vesicles come from to, join the Golgi? You have to know it's from the raphia. Okay, what do you guys think about this one? Should I launch the poll? You know what? I guess I'll just let you try it out, and then I will tell you the answers afterwards. But feel free to share if you have an answer. Nobody? All right, let's do the poll. Go for it. Only 3 people? Oh boy. That's okay. Well, don't be overwhelmed by all these words, right? Let's just take a look at it slowly. Once a solid material is phagocytized, that just means phagocytosis, right? It's been eaten up, basically, and taken into a vacuole. Which of the following statement best describe what happens? And the vacuole remains separate from the cytoplasm, and the solid material persists unchanged. Well, that's not true. If you bring something in, you gotta do something to it, right? You can't just let it hang around in the cell, right? What if you keep on bringing things in, and eventually you're gonna, overwhelm the cytoplasm, right? So that's not true. A ribosome enters the vacuole and use amino acids in the invader to form new proteins. That is nonsense. Ribosomes make proteins by using the messenger RNA, right? A lysosome combines with the vacuole and digests the enclosed solid material. That's correct. That's the answer. Okay, and this is also nonsense. Right? So like in the PowerPoint, right? Let me just show you. Lecture 3. Lysosome is down here. Right? You see, the food particle came in, the vacuole is going to combine with the lysosome, and it breaks it down, right? Okay. So all the informations are there. You just need to find the right place, I guess. Okay. Moving on. Explain what passive transport is. Provide examples of passive transport. So, I mean, when you see a question like this, right? Like on your own, you should also do active transport, right? So passive transport is from high concentration to low concentration, no ATP requirement. Example of passive transport would be like diffusion, right? For small nonpolar molecules, osmosis, facilitated diffusion. Okay, so see chart from lecture 4. Right, we again, it's a chart I showed you earlier, right, with the carrier protein and the channel protein, the one that we took the whole class to draw. So make sure you have that at your disposal. Now remember, you'll be using the lockdown browser, so any digital content on your computer will not be available to you during the test. So I suggest you, either print it out or even better, write one out yourself, right? I mean, you should have one already if you did it with me during class. Explain the differences between various type of bulk transport. Again, it's the chart. Maybe I'll just go back to the chart here and refresh your memory a little bit. So for bulk transport is all the way over here. That's a type of active transport, and we can either be bringing things in, endocytosis, or we can be sending things out, exocytosis. Okay, so that's the first difference. Are we sending things out of the cell, or are we bringing things in? In terms of endocytosis, depending what and how you are bringing things in, it's going to be different. Phagocytosis is bringing in solid, okay, like a bacteria, like a food particle. Pinocytosis is mostly liquid, right, extracellular fluid. And receptor-mediated endocytosis is, don't focus on the what you're bringing in. Right? In this case, is how you are bringing it in. Right? When you bring things in, it's like you can't just bring in random stuff. It has to match a receptor. So there's more control in terms of what is it that you are bringing in. Right? You have more selectivity in terms of what you are bringing into the cell. Okay, so those are the different kinds of bulk transport. Okay. Let's do another question here. Again, it's a lot of words, but don't get overwhelmed by that. Just read it part by part and see if you know the answer. Okay, I'm not gonna do the poll because only 3 of you answered last time. But I'll take it up in a bit. Feel free to share your answer if you have one. Okay, some people say B, some people say B or D. All right, let's go through it. Pinocytosis, which is the drinking, right? Drinking. And this one is the one where you have to match the receptor, right? So pinocytosis can concentrate substance from extracellular fluid, but receptor-mediated endocytosis cannot. Well, we are, both of them are bringing things in, right? So it's not about, concentrating it or not, right? So that is not the answer. Pinocytosis is non-selective in molecules it brings into the cell, right? It's just fluid. It doesn't really choose, like you might, as a Like when you bring in the fluid, you might accidentally bring in some other stuff. Think of it as like drinking from what's a good analogy? Drinking from a dirty pond or something. Like you use a straw and you drink. Yes, you're drinking the water, but you might bring in other impurities as well, right? Okay, you might bring in some dirty stuff along the way. Or like think about like drinking bubble teas. I don't know if you guys had that before. Like if you drink the water, you might bring in some some chunks of solid as well, right? So like it's not really selective. Whereas receptor mediated endocytosis offers more selectivity. Well, that's exactly what I told you, right? Earlier. Pinocytosis require energy, but this does not. Nope, they are both active transport, right? So both require energy. Pinocytosis increases the surface area of the plasma membrane, whereas mediated endocytosis decreases the plasma membrane surface area. What does it mean? Well, let's take a look here. This is the cell membrane. For pinocytosis, yeah, you're gonna bring things in, and then afterwards, you're gonna form a ball, and then you pinch it off, right? So this was part of the cell membrane before. Right? So after pinocytosis, you have less cell membrane, right? So it decreases the surface area, right? And that's the same case for endocytosis. So it should both be decreased. That's not correct. Pinocytosis brings in only water in the cell, but receptor mediated, and so endocytosis brings in other molecules as well. So like I said, it's not just water, right? Like it's called cell drinking, but there are so many stuff outside. You inadvertently might end up bringing in some solid as well. Okay. So this question, like I showed you, because I expect some of you see this, you might get a little bit confused because like they sound very similar, right? But hopefully after I explain to you, you understand why it is B. Any follow-up questions about this at all? No? All right. I am trying my best here to make this easier for you. Hopefully it pays off. Next one. The cell of a plant that grows in very cold environment. So we did this already, right? We did this already. Cold environment, right? If it's cold environment, you you want it to be less solid. So you want like more cholesterol, more unsaturated phospholipids, right? Fatty acids, right? And less saturated. Okay. How are active transport similar to facilitated diffusion? Okay, well, similarity. They are similar because they both required membrane protein. Okay, and they are different because this is passive transport. Okay, and for passive transport, no ATP is required. Now for lecture 4 here, there are still a lot of things that you, well, not a lot, but then there are still things like the hypertonic, hypotonic that we have not talked about here, right? But you should review that on your own. Okay, I'll give you a question to try. I will give you a question to try. There you go. Okay, we have B or D, we have A or D. Remember, you can only choose one thing on the test, right? So let's see what's going on here. This is placed in a hypertonic solution. Okay, so first of all, you have to, again, take out your notes or like, hopefully you remember it. Right? Hypertonic means it's more solute outside, right, than inside. So water is overall going to be leaving the cell. So A, the cell will show no change due to diffusion of both. So that's not true. It's not no change. The only time is no change is if it's isotonic, right? The cell will lose water and shrink. Now that sounds promising. The cell will swell and ultimately burst. That's for hypotonic. Okay, and then here, the cell will shrink at first, but later reach equilibrium with the surrounding solution and return to their original condition. That's not true, right? Once they shrink, they will stay shrunk. Is that the right way of saying it, right? Like they're not going to reinflate. The only time where if the water comes back in is if you decrease the concentration on the outside again. Okay, but if all else stays the same, once it shrivels up, it shrivels up. Think of a raisin, right? The raisin, once it shrivels up, it just shrivels up, right? If you put the raisin into a cup of water, then it swells back up to a grape. Okay, but like it's not gonna reverse itself on its own unless the environment changes, right? Okay, so I hope that is helpful. Okay, let's do another one here. I mean, let's move on to lecture 5 since we don't have much time left. What is an organic molecule? Organic molecule is basically anything with a C in it, right? Not sure why this question is here. It should be like in lecture 2, maybe. So any molecule that contains C, but excluding CO2, CO, and CN. Okay, so the people who came up with the rules, they're like, you know, let's make it confusing. Let's not count these for whatever reason. All the products of glycolysis. Again, I gave you a chart for that, right? So there's 2 ATP net, 2 pyruvate, and 2 NADH, right? Another suggestions I would have, like I mean, some of you, I don't know what you guys do actually, but like, you know, if you have your study guide answers in front of you during the test, I mean, fine. But it might even be better if you can take key things from the study guides, right, and reorganize it into a summary. Like you know, I'm gonna be asking you about the specifics of glycolysis and, you know, Krebs cycle and pyruvate oxidation. So, you know, one good chart or table to have is like this one, right? That's a nice one to have, right? So rather than flipping through all of them and finding it, you know, put it on a separate piece of paper, right? And there are a couple other charts in each lecture that are, you know, particularly useful to have. In cellular respiration, glucose is oxidized while oxygen is reduced. Explain what this means. So we know by now, basically, glucose gives off electron. Right? Glucose gives off electron. That's called oxidation. And then you have the NADH, FADH2, right? That will take those electrons to the ETC. They drop them off at the ETC, and then the electron get passed down the ETC, and then ultimately at the end we have the oxygen. Okay, so that's like basically the flow of electron. It's not a direct transfer of electron, but you know, ultimately it ends up on oxygen. This is called the final electron acceptor, right? Final electron acceptor. What is the role of NADH and FADH2? I just told you, right? They transport electrons that are released from the first 3 steps, and then they basically take it to the ETC, right? Where you can harness the energy and use it for cellular respiration. Sorry, use it to pump H plus across the cell membrane and generate the gradient, right? Next one. What is the significance of adding coenzyme A to pyruvate during pyruvate oxidation? So in pyruvate oxidation, 3 things happen, right? You remove the carbon dioxide, you produce the NADH, but you also add coenzyme A to re-energize, recharge the molecule, right? So that it will react further, in the Krebs cycle. Excuse me. All right, next one. Assuming mitochondria contains 50 NADH and 19 FADH2, if each of the energy carrier were used, what is the maximum number of ATP that will generate? That you will see something like this on the test. Why don't you calculate it quickly? Use your calculator. Anybody? Okay, 188. Is that correct? Anyone else? We did quite a bit of these, right? Hopefully we have to multiply 3 for NADH and multiply by 2 here. So that's 150 plus 2 times 19 is 38. You add them up, 188. Okay. The inner mitochondrial membrane area is 5 times the area of the outer membrane. What is the purpose of this? To maximize surface area, right? Such that more ETC can be packed in. Okay, the more ETC you have, the more places you can drop off the electron. Then you can pump the H plus over and create ATP more efficiently. Which step of cellular respiration generates the greatest number of ATP? It is the electron transport chain, right? So using oxidative phosphorylation, right? 34 ATPs, 32 rather, 32 ATPs are produced. All right. What happens to H plus as it is pumped into the intermembrane space? I mean, we talked about that multiple times already, right? This is the intermembrane space. You basically have lots of H plus here, and then that creates a high concentration. And then this is low concentration. So they want to come back in, but the membrane prevents them from doing so. So they have to go through the ATPase, which harnesses the energy and basically use the energy to produce the ATP. It's like a turbine, right? So that is it. And the last question, what is the final purpose of the final electron acceptor? That is to remove electron from ETC, right? So that the next electron will be able to flow, right? To flow past the ETC, so like you can continue the electron flow and continue to pump and create the gradient, right? Without the gradient, you cannot make ATP. Okay, let's just do one last poll question, and then we will call it a day. Anybody? What does it mean to phosphorylate something? Okay, Sarah says B, and that is absolutely correct. We wrote this down. Over here in our notes, right? I wrote it in pink. I don't know if you did. To phosphorylate a molecule means to add a phosphate to it, right? So literally, that's what it says here. A molecule is phosphorylated when a phosphate is added to it, right? Okay. The answer is B. Okay, bonus round, one more. I don't know if you guys remember the poison, okay? We talk about a bunch of poison that would disrupt the ETC, right? Rotenone, carbon monoxide, cyanide, DMP. Remember the article I showed you, right? The girl who bought the diet pill online and it turns out contained DMP, and she later was poisoned from it and died from it. Why? Why is DMP deadly? Okay. Nelson said D. Anyone else? All right, let's take a look at our poisons. Oh, that's not the poison page. This is the poison page. Right? Look. DMP is right here. Look at the picture. Right? What do you think is the right answer now? Oh, so many people choose D. But look at the picture. Right? Look at the picture. Instead of flowing through the ATPase, DMP makes it leaky, right? Makes the crystal leaky. So they just fall right back in without going through the ATPase, right? So the answer is A. Okay? That's it. All right. You still have time to study. I know there's so many midterms this week, but I genuinely hope that you guys do well in all of them. If you have any last minute question, feel free to email me. I will post this up online. Yeah, enjoy the week off. Next week, there are no classes. Next week is engagement week, and that will be it. Once you finish my test, I will not test you on anything that we've learned again. Right? We start brand new after the reading week, and that would be lecture 6. All right. So thank you for coming. See you guys next time. Have a good one. Bye-bye.